Okay, good morning, everyone. Thank you all for joining. My name is Arash Asher. I'm the Director of Cancer Rehab and Survivorship at Cedars Sinai Cancer. And I'm delighted to welcome you to One Bite at a Time, Emerging Insights in Survivorship Nutrition. We started this a number of years ago as a way to, spend, little windows of time diving into important topics that we think are relevant to either cancer survivorship or wellness in general. Megan, who I'll introduce in a second, has really been leading these on a variety of topics from, delving into some of the controversies of soy and breast cancer, understanding the role of nuts and seeds in cancer survivorship, mushrooms, etc. And we could share a link for the previous recorded programs. Today, we're extremely lucky to have an invited guest that Megan will introduce. Again, Megan Laszlo has been the lead dietitian associated with our survivorship program. She's been running and leading this series. She's been doing kind of oncology dietary work, I think about close to 15 years now. So we're incredibly lucky to have her experience both as a dietitian and as a chef, leading our nutrition and in our kitchen program. And she is going to introduce our guest today. So Megan, please take it away. Thank you.
Thank you, Dr. Asher. Well, I'm so excited for this collaboration today. I'd like to introduce Dr. Leon Fine. He has helped put this together and is the director of the Center for the Arts and Humanities at Cedars. One of their programs in the center is Food Lore and Culture, which has a seminar series dedicated to all aspects of food culture, ranging from science and medicine to nutrition to gastronomy to culinary arts. And the program has joined to sponsor this event featuring Dr. Suzanne Devkota, who is the first director of the program. Dr. Fine, would you like to say a few words?
Yes, thank you. We're delighted to join you this morning, and I hope that our mailing list of attendees at Food Law and Culture has joined in. It happens that the first director of the Food Law and Culture program was our very own Suzanne Devkota. So it's great to see her as the speaker this morning and looking forward to the talk. Thank you.
Thank you, Dr. Fine. Just a few housekeeping things. Please do mute yourself in this meeting. You're welcome to turn your video on. This meeting is being recorded without any of our participant names or photos. We do place our meeting recordings on Cedars Sinai Cancer YouTube, which I'll share the link in the chat as well. And I do ask that you enter any questions that you have that come up for you during our presentation into the chat so that we can review them at the end. We'll do a Q&A at the conclusion of the presentation. And I'll also put in our QR code for anyone that's interested in supporting our programs. Cedars Sinai is a nonprofit academic healthcare organization serving the diverse Los Angeles community and beyond. And our wellness, resilience, and survivorship programs are powered by the generosity of our compassionate donors. So I'll go ahead and share that in just a moment. But first, let me introduce Dr. Devkota. She is an associate professor of medicine and director of the Human Microbiome Research Institute at Cedars Sinai. She has been studying the effect of diet on gut microbiome structure and function in metabolic and inflammatory diseases for the past 17 years. She completed her doctoral and postdoctoral training at the University of Chicago and Harvard Medical School, respectively, and is an NIH Director's Pioneer Awardee and is a former Bronco Weiss Fellow of the Swiss Federal Institute of Technology. And she has joined us today to discuss the impact of diet on the gut microbiome. Thank you, Dr. Dakota.
Great, thank you. Thank you for having me. Uh, Both my parents finished their cancer treatments this year, and one of which was at Cedars. So it's really has this audience has a special place in my heart, and happy to speak with all of you. Um, Shall I go ahead and share my screen? Okay, great.
Yes.
Okay, and I did try to arrange this so we had enough time for questions at the end. I would love for this to be a discussion. So please, if anything here was unclear or you would like to learn a little bit more about anything I talk about, please ask me at the end of this talk. Okay. So the first part of my talk is going to be really an introduction to the gut microbiome. I know maybe we all are starting from a different knowledge base. Maybe we've heard of the microbiome, or maybe we haven't heard of the microbiome at all, but I kind of want us to all be on the same page. So part of the first part will be giving you just a primer on what the gut microbiome is and why it's important for our health. And then I will get into diet and different types of diet that affect the gut microbiome and some choices and guiding, decisions maybe that you can make in your own personal life. So, what is our microbiome? Well, the microbiome is not just bacteria, it's all microorganisms, all things we really can't see with our naked eye. I'm showing them here on the left. Our microbiome is bacteria, which is what you hear the most about, but also has fungi, yeast in there. There's also viruses. There's a lot of viruses in our gut, and many of these viruses actually uh, they infect bacteria. They're not necessarily ones that are infecting us, but they infect bacteria. But we also have unicellular organisms like protozoa. In some parts of the world, worms are part of the gut microbiome normally. So we have not just bacteria. There's many microorganisms in our gut, but the bacterial component is the most numerous of all of these species, which is why the field really focuses on the bacteria because they are what is most abundant. And all living species have a microbiome. I think that's important to note, ranging from corals in the ocean all the way to mammals, birds, fish, everything. And I highlight here our humble termite. The termite has a microbiome. It really consists of one organism. Its sole purpose is to degrade lignocellulose in wood. So sort of a form follows function in the microbiome. So every microbiome is adapted to what the host consumes and what the host needs. And that's quite interesting when you look at sort of comparative biology and ecosystems on Earth. So when we now talk about humans, that's what I think we're all interested in here today. And humans really are mostly microbes. And so if we put some numbers to that, what does that look like? So about 1 kilogram of our body weight is microbial biomass in and on our bodies. And that comes out to about 100 trillion bacteria. And so what the number that's been thrown around historically is that we are outnumbered 10 to 1. So for every human cell, there's 10 bacterial cells on our body. And that number's been sort of revised recently. And so at the very low end, it's maybe 1 to 1. For every human cell, we have one bacterial cell, which is still a lot. And at a genetic level, our human body has about 22,000 unique genes, but our microbiome and our gut microbiome alone has 8 million unique genes. And in the microbial genome, genes all have a function. And so when you think about all of these 8 million genes in our body, they must be doing something important. So where do microbes live in our body? Yes, our GI tract, our gut, has the biggest density of microbes in our body. So that's the largest microbiome. But there's also a nose, a nasopharynx, so nose and oral microbiome that's really distinct. There's a skin microbiome, which we've been hearing more and more about probably every time you open up some, app on your phone, you get some ad for some skincare product for the skin microbiome. There's also a urogenital microbiome, specifically in the female reproductive tract. The vaginal microbiome is very important for women's health. And so, if you kind of think about what is consistent about all of these, maybe except for the skin, is that these are all mucus-laden surfaces. We call them mucosal surfaces. So microbes love to live where it is warm and wet. And so on the skin, they tend to be most abundant in places like the back of your neck, the back of your ear, armpits, things like that. And what's pretty, remarkable about microbes is they self-assemble. So if you throw a bunch of microbes together in any environment, they will assemble in a sort of uniform way. So what I'm showing you here is an image of a dental plaque. So you go to the dentist and you get your teeth cleaned and they scrape, the little tool on your tooth, then it scrapes off a little piece of plaque. And so someone decided to take that plaque and do some staining of all the microbes that are in that plaque. And what they found, the image on the right shows it more nicely, is that you can see these bacteria that are colored in turquoise tend to aggregate in clumps more towards the middle. And then you see like the orange organisms and the green organisms more in the periphery. And what's interesting about that is that the organisms that are in the middle are the ones that don't like oxygen as much. So they tend to form at the core of any biological mass where oxygen doesn't penetrate as much. And the ones that can handle oxygen tend to be on the outside. And they tend to, aggregate in this way through, interbacterial communication, which is pretty remarkable. And this is true in all microbial systems in our body. So let's look at the gut. And so here's a beautiful images from one of my colleagues, Justin Sonnenberg at Stanford. And what I'm showing you here is first orient ourselves. On the left-hand side, here's, a cross section of your GI tract, a cartoon image. And the picture I'm showing you here on the right is what is captured within this red box here. So I'm showing you the inside, the big, what we call the lumen of your gut, on the upper left portion here. And the blue are your actual intestinal cells. So these are your intestinal villus. And the bacterial component is separated from your human cells by this really thick mucus layer, shown here in green. And you can see this mucus layer keeps everything on one side and all your cells protected from direct contact from bacteria. So I show this because yes, the microbes on the left, they are self-assembling in a certain way based on, the environment of the gut. But I want to visually highlight the importance of this mucus layer, because when you hear a lot about gut, leaky gut, you've probably heard about, which is a non-technical term, but we call it barrier disruption. What we're really talking about is a degradation of this green layer. And when that happens, the bacteria on the outside start to infiltrate your intestinal cells, and in some cases, can actually pass into your bloodstream or other tissues. And that's generally not a good thing. So a lot of our efforts, dietary and otherwise, is to maintain this gut mucus layer. So let's do a little bit of a history review for a minute on how we came to where we are today and appreciating microbes as this, co-evolved system with us. So it wasn't always that way. In the early 1900s, the focus was really on eradicating infectious diseases. So, everything you might, we take vaccines for, children take vaccines for. These were really what were killing people. It wasn't the chronic diseases that we see today. And so all the efforts were around maintaining and preventing infectious diseases. And that largely owed to vaccine development and hygiene, and particularly hygiene around our water supply. In parts of the world where this is still a problem, it's usually is due to inaccessibility to vaccines and contaminated water. So in the sort of more developed world, these are no longer problems really for us. And so what has actually happened though is we said, okay, well, we need to be, sanitary that prevents pathogens. So we kind of went the other way. We went hyper sanitary. And there was a period where everyone was using long before COVID, hand sanitizers actually had antibiotics, antimicrobials in them, if you can believe that. And so everyone's using them all the time without realizing that this low grade use of hand sanitizers was creating resistance, antimicrobial resistance on our skin. And then what you have seen today is a lot of blooms of these staph aureus infections that no antibiotics can treat. You probably heard of MRSA. That's an example. And obviously during COVID, there was a lot of, sanitization and that was necessary. But there's a period where that was extending, even before COVID to children, and everyone was hypersanitary. And so what has happened, there's this paper, really beautiful old paper for the New England Journal of Medicine. And on the left, they're showing time scales from 1950 to 2000 and showing how infectious diseases really dropped, as I showed you in the previous slides. And by the year 2000 in the developed world, not really a problem. But what in concert with the drop of infectious diseases came the rise of chronic diseases, shown here on the right. I mean, Crohn's disease, MS, diabetes, asthma, and so on have really increased significantly since the 1950s. And so one of the questions that comes up often is how clean is too clean? Is the over cleanliness been part of the reason, not the only reason, but part of the reason of the rise of autoimmune diseases? And in part, this is because we need environmental exposures. We need a little dirt in our lives to educate our immune system, especially in early life. And I'm going to talk about that in a moment. So there's been some of my colleagues have written these really interesting books, really focused on the early life microbiome in children, saying, hey, there is a period, where we don't let kids, if they drop it on the floor, don't let them touch it. And touching dirty animals and touching dirty things, you wipe their hands and oh my gosh, get that out of your mouth. But the pendulum is starting to shift, and because what has sort of happened is there's been a rise of food allergies and asthma and things like that, especially in younger populations. So there's a major movement in letting kids just be out there, and there is a strong link even between having a household pet and prevention of asthma. So these are things to rethink about, and the gut microbiome is really a big part of this. So we're now redefining our relationship with microbes. We've moved beyond this view of them as these external pathogens, and now think of them as maybe they're our friends. Maybe we want to support them, and because maybe they're actually doing something important for us. And how do we distinguish these good ones from the bad ones that we don't want? And so some really important themes have come out of this. I've talked about what's called the hygiene hypothesis, this hypothesis that over cleanliness has led to a rise in chronic diseases. Kind of in concert with that, we're rethinking antibiotic use. Overuse of antibiotics for a cold, for a flu, for things that are inappropriate for antibiotics. There's a period where people took them like candy. Now, in some parts of the country, it still is the case, unfortunately. But certainly in California and LA area, I think there's been a major trend in people questioning, do I have to take this antibiotic use? And I think a good example is if you go to the dentist and you're going to have a root canal, you're usually automatically prescribed a prophylactic antibiotic. And I know from personal experience when I asked my dentist, I said, do I have to take this? And they say, well, no, you don't have to take it. And so you should always ask, do I have to take this antibiotic? And if your doctor says yes, then do it. But if it's optional, then you may want to reconsider. Questions around early life seeding of the microbiome. How do we get our microbiome in the first place? What is appropriate, seeding of the microbiome? How does breastfeeding versus formula? How does vaginal versus C-section? How does all of that affect our normal colonization of our gut? A big focus in the field is on disease linked to microbiomes. A lot of research into the role of the microbiome in cancer and tumor biology, and also responsiveness to anti-PD-1 therapy and things like that. So there's a lot of really interesting work coming out of this space. Certainly in inflammatory bowel diseases, Crohn's and colitis and colorectal cancer, a really hot area, and also in cardiovascular disease. And kind of linked to this, we really are studying environmental perturbations. What is in our external environment that is perturbing our microbiome and maybe shifting it away from an ideal state? And are there things we can do to shift it back? And what is that? What is the ideal microbiome? So now let's start to talk a little bit about diet. And I think what is really important to note is there's so many environmental factors that affect our gut microbiome. But in the microbiome field, it is uncontested that diet is the primary driver of our gut microbiome composition. The only other thing that influences our microbiome more is antibiotics, which is obvious, but that's not something that we chronically take in our everyday life. Diet is something that we encounter every single day. We have to. And our gut microbiome is largely determined by the diet that we eat and a little bit of our own body chemistry. And so this image that I'm showing you here, this little cartoon in the middle, showing you the small intestine. So your GI tract, at least your lower GI tract is your small intestine, which is where most of your vitamins and dietary nutrients are absorbed. And then I show you the colon, and the colon is largely for water reabsorption and for fermentation of foods that our bodies can't digest. And how does that happen? It happens through your gut microbiome. So microbes that colonize your small intestine are really tailored to the functions that go on in your small intestine. They might be contributing to the nutritional milieu of your body, whereas the microbes that are really efficient at fermentation, they live in your colon. And this sort of shakes out naturally, which is pretty interesting. And what does this actually do for us? So, in a practical sense, what do we know in the field, and what has been studied in terms of colonization of our small and large intestines? Well, the microbes, when they colonize there and they interact with our bodies, they produce, just like we take in oxygen and breathe out CO2, they take in various chemicals and they breathe out various chemicals and gases as well. And we study those and we study those effects on the body. And what we know is that these byproducts can relate to our immune system homeostasis. How reactive is our immune system? It can interact with our central nervous system because our GI tract is highly innervated. It's one of the most innervated organs in the whole body. They produce vitamins for us that we can't get any other way. Vitamin K, certain B vitamins come from our gut microbiome. They detoxify drugs, they make antibiotics, they actually interact with hormones like estrogen and androgens. So they do a lot of biotransformations of things that go on in our bodies. And so this really highlights how important it is to understand what they're doing and how to support them so that they're doing the right things and not the wrong things. And so, yes, if we don't support them properly, they can lead to different types of diseases, either primarily in the case of, ulcerative colitis or Crohn's disease, or they can be secondary or tertiary, synergistic responders along with some other factor. But if you support them, they can also support optimal health. And I have to say, there's still a lot we don't know about either of these states in the microbiome field. There's still a lot to be learned. So when we think about colonization of our bacteria in our gut, I mentioned that, they colonize where their function is most useful. Um, so so it really is about form following function. If the function is needed, that bug colonizes there. It's not the other way around. But in the disease state, when you have a perturbation of your microbiome through antibiotics, through uh, international travel, through a radical diet change, through pelvic radiation, things like that, the rules of engagement in your gut microbiome change, and it becomes about survival of the fittest. And so the microbes that are most fit to handle these harsher environments tend to be the ones that survive. And often these microbes that are quick to evolve and adapt to these perturbations tend to not be the most favorable bacteria. And so there's a lot of focus on how do we then, support the microbiome during times when we can anticipate a perturbation. So let's go to talking about how do we get a microbiome in the first place. So the gut microbiome is really seated in the first 3 years of life. The placenta is sterile. Babies do not have a microbiome when they're in the womb. Their first exposure to a microbiome is really through the birthing process. If it is a vaginal birth, their first microbiome exposure is mom's vaginal and stool microbiome. Because during the birthing process, everything comes out and babies get exposed to all of that. If a baby is born by C-section, their first microbiome is largely a skin and environmental microbiome that comes from that operating room. So C-section or vaginal, those are questions we ask. Then how is the baby, nutritionally supported after that? Are they breastfed? Are they bottle fed? Is it a hybrid? That can also affect which microbes colonize the baby's gut as well. And then what environmental exposures does the baby see in early life? Where in the world do they live? So their wean start to see, native foods around 6 months of age and start getting more and more food exposures. Well, if you live in LA, you'll see one kind of diet. If you live in Africa, you're going to see a different kind of diet, and that will affect which microbiome colonizes that gut at that time. Does the baby have a lot of ear infections and has to go on antibiotics a lot? That will affect microbial colonization. Are they exposed to viruses? We know in early life, viruses have a bigger impact on the gut microbiome than they do in later life. These are all things that we ask. So this is a figure, and it looks complicated, but it's actually really fascinating. I'm going to walk you through it, but I show it in almost every one of my talks. This is the gut microbiome of one baby in the first year of life. And along the x-axis down here are different life events that the baby was exposed to during this period. And each black hash represents a unique microbial species. So let's look at this period right here. This is when the baby was exclusively on breast milk. And all I'm highlighting here is that there's a certain number of black dashes that represent the microbiome that is adapted to breast milk in this particular baby. Nothing, nothing really out of the ordinary here. But now the baby starts to have a hybrid of rice cereal and breast milk. You can start to see the types of microbes that colonize now are slightly different. You see the density of these dashes are in this region, meaning that they're starting to lose some of these breast milk microbiomes down here. The ones that were here are no longer there. And they've acquired some new ones up here that weren't really present much before. Now they're transitioning to like adult table foods. Now, in this period, you really see a loss of all of the breast milk adapted microbes. Some still persist, but largely they're gone. And now they're starting to shift into what maybe is looking like a more mature microbiome. And now the baby's on a full adult diet, eating what largely what their parents are eating. And you can see they now have a completely different microbiome than they did when they were first born on breast milk. And this is the normal succession of the microbiome. And this microbiome right here will largely persist throughout this infant's life. So I want to kind of zoom out a little bit because there's some important points to highlight about diet at the population level versus the individual level. And I'm going to start with talking about diets across the world because we actually have the most data on population level microbiomes. And so what I'm showing you here is with these asterisks are all the studies that have looked at populations in these different regions around the globe. So we actually have a fairly wide coverage of different regions of global, parts of the world represented in our microbiome analyses. Now, some of these have very small numbers of individuals in them, and some have a lot more, so it's slightly skewed. But we do have some sense of how microbes differ around the world. And of course, the layer below that is what is the reason for these differences, because there are major differences. So this study was really fascinating, I thought. It's from 2012, but still so relevant today. And what they did was they took microbiomes across the lifespan in these three distinct regions of the world. Okay, here in the US, this is actually St. Louis, in the Amazon region, and in Malawi in Africa. And so what they show here is, so in this first figure, along this bottom axis is age. So every dot is one human microbiome, one individual person. And you can see that there's, some individuals that were 82 years old represented here and a lot of babies. The y-axis represents how many unique bacteria do they have. So the higher up you are, the better it is. You want a lot of bacteria. That's a good thing. We call that diversity. And diversity is directly associated with health. So when we see loss of diversity, that tends to mean a loss of a healthy microbiome. So two things, and in blue, that's the Americans. Amazonian from Amerindia were in green, and Malawi are the red dots, individuals from these regions. So one important thing is the samples that came from babies who were still breastfeeding or bottle feeding or living very close to their mothers, their microbiomes all tracked. They look no different no matter where in the world you lived. So as long as the babies are on some form of breast or bottle feeding, microbes tend to develop the same. It's only when at this age when they start to really shift to their native diet, they're weaned onto whatever diet is local to their region, you start to see a separation. And what's very clear is that the United States microbiome is far less diverse than in the African or the South American populations. And these are two different continents. It's really remarkable, but their lifestyle practices are much more similar. When we look at this figure is showing now how similar are the types of species within these microbiomes. So what you can see is not only is our microbiome less diverse, we also have entirely different types of bacteria that colonize our gut. Whereas here on two different continents, their microbiomes look much more similar. So it's not about, environmental reservoirs. These are microbes that live in those regions. What is really, the cause of that? In a separate study, this group took a large population of looking at sort of more ancestral hunter-gatherer stools that they acquired compared to industrialized, these would be American populations, and looked at the different types of bacteria. And so all this is simply saying is that, hey, we have different microbes than our hunter-gatherer ancestors. Then they took a population from within the same country in modern day and looked at a city dwelling group of individuals versus individuals that lived in remote farming regions. And we're talking modern day and just different lifestyle practices, and we have different microbiomes. And one of the things they also found in this study is that because I mentioned that having a diverse microbiome is equated with health, when they looked across all the populations at the most diverse microbiomes, they looked at what individuals were eating. What stood out the most was that the individuals with the most diverse microbiomes were eating 30 or more different plants in their diet. So this is not saying we're talking about just being vegan or vegetarian, but the diversity of plant matter really supports a diversity of gut microbes. And that's a really interesting concept to think of as you maybe think about your own diet. How many plants are you eating? It's not just, we're not talking about leaves of spinach, we're talking about grains, quinoa, things like that. So it might be worth counting in a day how many different plants are you eating to support your optimal diversity. So, in a similar population study, they wanted to compare the within a 1 year period how the microbiome shifts over 1 year in a more Western population compared to the Hadza, which is a hunter-gatherer tribe in Tanzania. This group has been well studied in the microbiome literature. And what's really remarkable is they found in the Hadza, who really eat off the land, they don't have the grocery stores, they're really eating what is available at that time, their microbiome cycles with the season. So in the wet season, they have a different microbiome than they do during the dry season, but it goes in this predictable cyclical pattern. And as you could predict, it's largely due to the food that is available during those times. And here I'm showing you an image of the types of grains and tubers and things that they grow. These are staples of their diet. I don't think any of us eat anything that really looks like this. And their microbiome is considered one of the most diverse microbiomes there is. So they extended this to, look at these trends across more populations. And so they took other quote unquote non-Western populations here and compared to some Western type countries, Japan, China, UK, US, et cetera. And they clearly see a trend that in non-Western countries, diversity is just much higher than it is here in the Western world. And again, the conclusion of the study is that food availability is very different here in types of foods that we have, but also we don't really eat seasonally here. We have completely moved away to seasonal eating, and we eat strawberries whenever we want strawberries and so on. But in these other regions, they really eat seasonally. And is that maybe the way that our bodies and our microbiomes were adapted to eat? So, in a similar but different large-scale sequencing study where they looked at the microbiome of many other, of other populations, urban hunter-gatherer, some additional things that they have found is that the microbiome in our Western world actually carries more antibiotic resistance genes than they do in other parts of the world. And you can imagine that, in other parts of the world, they're just not using antibiotics the way they do, so they don't carry around these resistances like we do. And antibiotic resistance genes, they get passed on to other bacteria in our body, and so they don't just stay within one microbe. They get passed around and spread. And as you can imagine, that's not a good thing. And so here's some other examples of studies that have been done that have found all similar things. And what all these studies have concluded is that the move away from seasonal eating in our more ancestral type of diet has resulted in a loss of bacterial diversity, increased antibiotic resistance genes, and also a greater incidence of chronic diseases. In these other populations, chronic diseases really don't exist. And one other kind of fun thing that has been reproduced over and over again is that also, and it's kind of related to lifestyle factors, is close social relationships correlate with human microbiome composition. So what does that mean? If you live in the same household with someone, your microbiome will be more similar to them than it will be to your neighbor or someone you work with. And this has been proven time and time again. Actually, in the old days of fecal transplants, which is a therapy for chronic C. diff infection, now they have pills you can take, and it's much more standardized. But before, the recommendation was if you come into the clinic and antibiotics aren't working and you have this chronic infection, fecal transplants were the most effective therapy. And what the recommendation was your donor should be someone you live with, because they will be most representative of a microbiome that will actually colonize you because you cohabitate. And that's a really interesting concept. So in this paper, they took a bunch of couples. Okay, so I show here they took 94 spouse pairs, so people that are cohabitating and presumably in intimate contact, and 84 sibling pairs, so people who might live together but aren't as close as spouses may be. And they controlled for all of these factors. So sex, blood panel, all that was controlled for computationally. So those are not factors in the results. And they looked at first here on the left, just microbial diversity. Remember, I said diversity is associated with health. And if you're lower on this graph, it is actually more similar to each other than different. Okay, so you want to be lower on this graph. And spouses had a more similar microbiome to each other than siblings did, and certainly than unrelated people did. And then they grouped people they surveyed them and said, okay, how intimate, how close would you say you are? And the spouses that said they were very close had a much more similar microbiome than their not so close, partners. And then they went on to kind of compare this across now siblings, asking the same thing. And spouses always had a more similar microbiome than siblings did. But siblings who said they were close and they probably hugged a lot and so on had a more similar microbiome than the siblings who said they were not very close. So when you think about now, these populations around the world that have more diverse microbiomes, and they share their microbiomes a lot more because they eat communally. And that's a big thing that we have lost a lot with industrialization in our modern lifestyle. So something to think about in terms of sharing microbes is actually a good thing, and that has been associated with health within a household, sharing your microbiome. So to summarize that part, at the population level, we clearly have distinguishing gut microbiome compositions, and they're almost solely attributed to distinct dietary patterns, and that's somewhat conferred by the way in which we eat together. And the most studied comparison is Western, non-Western, industrialized versus non-industrialized. And these terms are very sensitive terms. There's a lot of issues around the terminology we use around industrialization. So I'm not claiming that these are black and white definitions. But I think you understand what I mean when I, describe these in relation to the previous studies. But at the individual level, if talking about me versus you, the same diet often doesn't lead to the same microbiota composition or functional response. We're as unique as snowflakes. So you need really large population sizes to see these differences. But when you get down to comparing me versus you, there is a lot of variability there. So let's talk about microbes at the individual level. And really, I'm just going to talk about one study because this was in 2015. This has to date been the only study that has prospectively done dietary interventions and measured various responses to different diets to actually understand how inter-individual we are. And what their primary output here was they were looking at glycemic responses. How much does your blood sugar spike after eating a given food? And they measured all of these different factors in response to the dietary intervention. They looked at stool microbiome, blood tests. They asked questions about lifestyle factors, took body weight, height, all that sort of stuff. So they had all of that information, but they're really looking at glycemic response to different foods. And here's just a schematic of the study design, really rigorously designed, beautiful study of 800 participants, probably a very expensive study as well. And here's an example. Here's a figure from this. We're looking at blood glucose. So here on the left is blood glucose levels. And they're taking an example of 4 individuals from this study, participant 67, 663, and so on. And in response to eating a defined amount of bread. And very clearly, you can see that the individual response to eating the same piece of bread is very different. This individual right here almost had no glycemic response to eating bread, whereas this person had a huge glycemic response. Now they take an example of here's participant 468, who is given bread or glucose, which glucose you would expect. I mean, the biology would say glucose should spike your, blood glucose in your blood. This person had a high response, as you would expect, to blood glucose and a normal response to bread. Participant 663 had a high response to bread, not nearly as high a response to glucose. Really interesting. Now they compare banana to cookies, which you would expect to have, a different glycemic response. Participant 445 had a huge glycemic response to bananas and almost no response to cookies, while the opposite was true for this different participant. So how we eat, we're each very unique in our response. And they also went on to look at sushi and hummus and test all these different foods that we, associate with health. And they found that some people had a very high glycemic response to hummus, but not to bread. And these are, really fascinating. And they tied several of these associations to gut microbiome changes. And they actually created a company where you could test your blood glucose and your gut microbiome and make some food recommendations. And so, anyways, just highlighting that inter-individual differences are very high, which makes it really difficult to study this and determine what is the best diet for each person. The reality is you have to listen to your own body. So now let's get to some practical things. What are some, universal things that we know from, decades of studying we meaning the field, decades of studying diet and microbiome interactions? So ultimately the goal is to promote the growth of bacteria that make what are called short-chain fatty acids. Okay, and I didn't go into much about the details of short-chain fatty acids, but essentially these are the byproducts of bacterial fermentation of different fiber sources in your colon. And short chain fatty acids are made up of three, molecules right here. And these are the primary energy source for your gut cells. Your gut cells really love short chain fatty acids. It keeps them happy and it keeps them strong. Excess short chain fatty acids, which is very normal, goes into your bloodstream and is actually elicits effects in your immune system. Good effects. It stimulates actually T regulatory cells, which won the Nobel Prize a couple days ago. Stimulates liver, pancreas, and fat tissue in some beneficial ways, and also interacts with your central nervous system. So why are these important? What I just mentioned. So more specifically, they maintain your gut barrier. So because they feed your intestinal colonocytes and keeps them happy, it keeps this barrier intact. Okay. If the barrier is not intact, things that are flowing through your intestines will start to leak into your bloodstream. That's microbes themselves, antigens, food products, chemicals that should stay in your gut start to seep into your circulation and will cause an immune response. And so really focusing on maintaining gut barrier is really key for systemic health. And you do that by promoting these short chain fatty acid producing bacteria. And many aspects of our daily life can transiently affect our gut barrier. So, sleeplessness, poor diet or radical dietary changes, stress, medications, all of this can transiently affect our gut. This is actually pretty normal and will largely happen if they're in, periodic episodes without little consequence. But if these things become chronic, then you can start to, have a slow burn, a slow leak leading to a chronic condition. So you've all heard about prebiotics, probiotics, postbiotics. I think that's what we all want to know about here. What's the difference? What should I take? So prebiotics are foods that we eat that support your gut microbiome. So these are not, taking the bacteria themselves. This is how do I support the organisms that I already have in their most best expression of themselves. Probiotics are what you see hundreds, thousands of them on the shelf, advertised to no end. This is taking bacteria in a capsule and hoping that they will colonize your gut and confer some benefit. And then postbiotics is a much newer concept where this is actually the chemical byproducts that bacteria make and finding a way to take those directly for health. So that's like, what if I wanted to take a short chain fatty acid directly? Can I do that? So let's talk about prebiotics, feeding your good bacteria. Um, what we know is that various things, as I mentioned before, but chronic inflammation, medications, if you've had pelvic radiation, this will suppress your good bacteria, but rarely are they gone for good. Rarely are you extinguishing your microbes. They just might be reduced or suppressed. You can totally revive them through dietary prebiotics, and these are largely fibers. So certain fibers specifically target, though, those short-chain fatty acid producing bacteria that you want. So what are some examples of these fibers that you can find in food products that you may want to keep an eye out for? So inulin, asparagus has inulin, chicory root, pectin, this is from the skins of apples. So don't juice your apples, eat your apples. The skin's really important. Resistant starch from green bananas, beans have resistant starches, and beta glucans, so barley, mushrooms, and so on. These specifically feed those good bacteria that produce short chain fatty acids. And while I don't promote products, that's not my thing, but I did come across this product at Whole Foods the other day that had a really, I thought, really excellent profile in terms of prebiotic fibers, low sugar, high protein, and so on. So I started consuming this for morning for breakfast. If you find this, I found it at Whole Foods. This has a great source of fiber, very low sugar, 20 grams of protein, 100 calories per bottle. Really great way to start to add something to your diet that helps your gut microbiome. Now, probiotics have fallen out of favor in the microbiome community. So why is that? It's still unclear how well a consumed probiotic actually colonizes. So you take it, but the formulations either don't have as many live bacteria as they advertise, or if you may already have that bacteria in your gut. If it already exists in your gut, you won't colonize if you take it in a pill. So you could be just wasting money. So it's still unclear in a given individual if these actually are doing anything. As I mentioned, there's inconsistencies in probiotic formulations. What has been shown is that there's been these large scale analyses where there's really little benefit in adults, especially this has been studied in adults with GI conditions, but there does seem to be some benefit of probiotics in children. Either healthy or children with GI conditions. And that's largely because their microbiome is still very nascent and forming, and there aren't a lot of bacteria in there. So if you take a probiotic, they actually might colonize. In adults, we have so many bacteria already that's really hard for these pills with a foreign bacteria to come in and colonize. Now, one thing to note is that patients currently undergoing treatment, or if you're on immunosuppressive therapy, you really should avoid probiotic supplements. So it really comes down to, prebiotics. That's the safer, gentler way to actually support gut microbiome growth. It's the natural way. Feed what you have, bring them back to life. They're still there. They're not gone. But you have to be really patient. And postbiotics, consuming the beneficial chemicals bacteria make, this is a really emerging field, and you're going to hear a lot more about this in coming years. You probably hear a lot about fermented foods. Fermented foods probably have the highest abundance and density of these beneficial metabolites. So you can't take short chain fatty acids directly in a pill. If you see them advertised, I wouldn't believe it, because if you take it through your mouth, they actually get absorbed very high in your GI tract, where they don't confer the benefit. So be wary of those. But it's also still unclear. So what's the dosing? How many of these chemicals, postbiotics, do I need to take to have a benefit? We don't really know that. But examples of the types of metabolites that are in something like a fermented food, there's B vitamins, there's antioxidants, there's acids, even serotonin. We found that in kombucha, and acetic acid, which is good for gut health. So fermented foods, if you can tolerate them, are really rich in postbiotics. So combining with a prebiotic fiber, taking fermented foods can be a really nice way to support your gut microbiome. Oh, one thing to note is that individuals, if you're on an MAOI, you should avoid fermented foods because there's an interaction there. Okay, finally, recommendations for long-term gut health. First and foremost, if you're on treatment or going to be anticipating any kind of cancer treatment, first focus on nutrition to support whole body health. Worry about your gut second. So protein, energy needs, take care of that first. After treatment, then you focus on restoring microbiome balance. Think about different dietary interventions, but you have to think about your tolerance. Not everyone will be able to tolerate the same types of fibers or fermented foods. If your gut hurts, don't keep taking it. You got to really spend some time listening to your body. As I mentioned, if you're on MAOI, You should avoid fermented foods. If you have concurrent IBS, which may come as a result of treatment sometimes, which is irritable bowel syndrome, you may not be able to handle all fibers. And so there are these FODMAP diets you may have heard of, which is an avoidance diet that can help you start to restore your gut microbiome and then slowly add these fibers back. But it's an elimination diet first. That's only if you have IBS. And lastly, be patient. Diets take time. Usually, it takes about 4 to 6 weeks of consistency to see a measurable effect in whole body dietary, effects, but also in your gut microbiome. Some things to pay attention to that I advocate very strongly is pay attention to stool consistency, frequency, color changes, etc. All of these can be early signs of either, a good shift in your microbiome or potentially other problems. So while it may not be the most pleasant thing to do, there's a lot you can learn from this. And I will end there with any questions.
Thank you so much, Dr. Devkota. That was a fantastic presentation. We're a little over time, but I want to make sure we have time for a couple questions.
Sure.
I'd like to start us out. We've done a great job discussing how what we eat affects our gut microbiome, but to what extent, if any, does our gut microbiome affect what we eat or influence our food choices?
Yeah, that's an emerging kind of area. People who are studying the gut-brain connection, for example, it's really inconclusive how um, the microbiome affects food preferences. That we're not really sure about. And there probably is not so much a driver there. Now, the gut microbiome could affect satiety, so how full you feel, through the production of these short chain fatty acids, because they actually confer about 10% of our caloric needs, this production. So if you're deficient in certain short chain fatty acids, you might have this drive to eat more. But that's all largely still in the realm of associations. We don't have mechanisms for that yet.
Thank you. And then we have a question of if a strong antibiotic is unavoidable, how best to rebuild the gut microbiome at the end of that treatment?
Yeah, great question. So, what we recommend now is you really want to focus on dietary diversity during that period after antibiotics. Don't try anything while you're taking antibiotics just because you're going to be depleting your microbiome anyway, right? But after you finish your course of antibiotics, you want to focus on, remember I said that individuals who ate 30 or more plant products had a more diverse microbiome. So really focus on bringing a diversity of plants into your diet, and see how your body handles that. But that will really help support that microbiome reconstitution.
Thank you for that. Dr. Fine, I see a question from you.
So yes, so my question, Suzanne, is, there's one of the hypotheses for the evolution of Crohn's disease is that there's some systemic deficiency in phagocyte function in leukocytes. And so my question is, to what extent is the innate immune function of the gut possibly a conditioner of the response to microbiomes in various disease states?
Yeah, it's certainly possible. I mean, there's people studying this. I mean, we mostly study the microbiome reaction with T cells, with the adaptive immune response, and particularly around regulatory T cells. There isn't quite as much known on the, innate immune system, but clearly that's involved. But I can't really point to any definitive studies where they found like a key microbial culprit that might be involved in this response, certainly in Crohn's disease. But there's certainly a there because that's what the um, ability for our innate immune system to detect bacterial invasion into the gut and so on is key. So if you're deficient in that, you could imagine things related to sepsis and so on. But it's still an area for exploration.
Thank you.
Well, I do want to be respectful of everybody's time. I so appreciate your time and attention and this meeting and sharing your research with us. Thank you so much. Dr. Asher, do you have any concluding remarks?
No, I want to thank Dr. Devkota so much. We obviously there's a lot of questions we could not get to, so I apologize for that. We will continue with this series, and maybe we'll be able to revisit this in a different format next time. Thank you all so much for attending. We look forward to seeing you for the next seminar.
Thank you.
Thanks so much.